Hey guys, this is me Akash here for Biology Vibe. So today we're gonna start a new Vibe 1.5 wherein we're gonna discuss about the replication of DNA. So replication, as the term suggests, directly basically copying a single strand of parent DNA molecules to two identical DNA molecules. Simple basic definition of DNA replication. Further, the question is, why is DNA replication important? So mainly, when the cell divides, the DNA has to divide with it. Otherwise, problems would occur. Problems like, just suppose that if a cell divides and the DNA doesn't divide, so the daughter cells will get half the amount of DNA or a slight less or a slight more amount of DNA. If the DNA divides and the cell doesn't divide, then the same cell will have the double amount of DNA it should contain. Then also it's a problem. So the cell division and the DNA replication should be highly coordinated. And that's important because the DNA is the actual genetic material and it has to pass on the genetic information which is important for the survival of the cell. So it was about what DNA replication is and why it is important. So let's have a look about the machinery which is there. So DNA replication basically requires a set of enzymes and it's energy dependent. Energy dependent, that means it's utilizing energy. It's an active process. It takes place in the S phase. Cell cycle mainly has, if you have studied in junior classes, like G1 phase, S phase, G2 phase and M phase. So in the S phase, that is the synthesis phase, the DNA replication takes place, and it has the cell division and the replication of DNA should be highly coordinated. So the process requires parental DNA. Definitely the DNA which has to be copied should be there, the parental DNA. DNA dependent DNA polymerase and enzymes basically which depends upon the DNA, and it takes up raw material from the nucleoplasm and adds up to make the new DNA. RNA primer, DNA ligase, topoisomerase, all these enzymes are involved in this machinery. Where they are involved, why are they important? Let's start with that. So this is somewhat a typical structure of a DNA replication looks like. So first of all, it takes place in alkaline medium. The process starts at ori, that is origin of replication. The replication process does not start at random at all. There are specific regions from which it will start. Those regions are called ori. What happens is, like, topoisomerase enzymes induces a cut in one strand of DNA, that is called nicking, to relax the strands of DNA. Basically what happens is, like, this is a double stranded DNA. Hydrogen bonds are being broken from here. That means it's opening, and when a supercoiled structure is opening, definitely it will cause strain at the back regions. So topoisomerase basically enzymes, what it does, that it cuts that one strand of DNA and then relaxes the strand so that DNA damage does not occur. If a supercoiled thing is opening, definitely it will cause some strain at this region. And this topoisomerase enzyme basically works to reduce the stress. The separation of two chains of DNA is done by breaking hydrogen bonds, and these hydrogen bonds are broken by helicase enzyme. Okay, this helicase enzyme is responsible for breaking these hydrogen bonds between the nitrogenous base pairs. Now, definitely, if the bonds are breaking and strands are going single, so helix destabilizing protein prevents so that recoiling, that is attaching back again formation of hydrogen bonds, it prevents that. And secondly, SSB protein, that is single stranded DNA binding protein, just sticks to it the single strand and it binds to that so that it's a bit stable. This single strand is bit stable so that there are no bends or loops in this strand. So till now we have studied about helicase enzyme. The helicase enzyme is responsible for breaking the hydrogen bonds. SSB proteins attaches to the single strands to stabilize that so that there is no loops or bends. The topoisomerase enzyme is basically responsible to reduce stress due to superbinding. So now the replication starts. So first thing which is to be noticed that the structure looks like a fork, right? So this is known as replication fork. One more thing which is to be kept in mind that the replication that the DNA polymerase will start synthesizing the new strand of DNA but only in one direction, that is 5 prime to 3 prime direction. And we all know that the two strands of the DNA are anti-parallel. That means if you look at this strand, the synthesis is quite simple. This is known as a leading strand. This is 3 prime, so on the opposite 5 prime should be there. 5 prime is there. DNA polymerase is just taking up the raw material, that is the nitrogenous bases, sugars and phosphate groups, and adding it up here. So this basic extension of this 5 prime to 3 prime end, new strand is being made. But the problem is for the other strand. Now what's the problem? It has that is basically the polarity issue. The DNA polymerase will not synthesize from here to this side, right? So what happens here? RNA polymerase, which is responsible for synthesizing the RNA primer, that is small segments of RNA are attached here. So that RNA primers provide 3' OH end to the DNA polymerase, and then the DNA polymerase extend this region. Definitely it will lead to some discontinuous replication, right? Some small fragments are being built up. Now these small fragments are known as Okazaki fragments, part of the lagging strand. So this is known as the leading strand and this is known as the lagging strand. So formation of new chain is always from 5 prime to 3 prime direction. One chain is continuous, this is known as leading strand. The formation of other strand begins from the center and not from the terminal end. It will not start from here, rather it will start from the center in this direction, 5 prime to 3 prime, this direction. Chain is discontinuous, made up of small fragments known as Okazaki fragments. This is termed as lagging strand. Okay, and then what happens? Small spaces are there, and this is joined by DNA helicase. One more enzyme is there. The formation of new chain is catalyzed by the enzyme DNA polymerase. Okay, so this is a kind of a whole diagram what things happen. Let's start with a quick summary of what we have learned till now. So the first thing is like this is the parental DNA. First thing is enzymes unwind parental double helix. This is done by the helicase enzyme, right? So helicase enzyme is basically responsible for breaking this hydrogen bonds. It's breaking, right? Now the second point is protein stabilize the unwound parental DNA. This is done by SSB protein, single strand binding, that stabilizes the single strands and prevent it from again coiling it back. Now the third point is the leading strand is synthesized continuously by DNA polymerase from 5 prime to 3 prime direction. DNA polymerase is synthesizing. Now the next thing which comes into picture is the lagging strand is synthesized discontinuously. RNA polymerase synthesizes a short RNA primer. So this RNA polymerase is there. It is responsible for the formation of this RNA primer, that is this blue segment, small segment, which is then extended by DNA polymerase. So DNA polymerase then comes into picture in the lagging strand and it extends the RNA from the RNA primer. And then what happens? The DNA polymerase digests RNA primer and replaces it with DNA. So this is a little bit tricky portion. You have to understand that when firstly the RNA polymerase comes, it synthesizes RNA primer. When it synthesizes RNA primer, this will assist DNA polymerase. At last it will give a 3' OH end to this DNA polymerase, and this DNA polymerase will extend the chain, and then DNA polymerase will chew up this RNA primer and extend the DNA. Now this replication is done in form of fragments. This is known as Okazaki fragments. At the end, DNA ligase is responsible for joining the discontinuous fragments of the lagging strands. So that's kind of it, guys, and I hope this video helped you. In the next vibe, we'll start with the central dogma of biology, and we'll discuss a little bit about the genetic code. Thank you for watching. If you like the video, please hit the like button. Definitely share it with your friends and do subscribe to my channel to get more and more videos like that. Thank you.